introduce Dr. Matt Forister. So Matt is not only an accomplished scientist, but a pretty remarkable mentor, teacher, and community member, both within and beyond academia. We're really lucky to have him visiting us. We encourage all of you to chat with Matt. If you don't have a one-on-one lined up, we have our social after this talk. So do make time to chat with him. So Matt received his Bachelor of Arts from the University of San Francisco. He majored in English and reading classics by Gould and others. He was drawn into the big open questions in ecology and evolution. Matt was mentored in graduate school by the consummate natural historian, Dr. Art Shapiro at UC Davis, and postdoc with a foundational thinker in evolutionary biology, Doug Kuchuma. Matt is now carrying the torch to inspire a new generation of scientists. Since beginning as a professor at UNR, Matt has received numerous awards, including selection in 2015 and 2020 as among the best professors in the entire university across all the departments. His research, it's a long block of words. His research has incredibly broad impact. Just check out his scholar page, and has advanced our theoretical understanding of insect diet breadth evolution, helped create practical conservation plans for many Western butterfly species, raised awareness of threats facing native insects at local and global scales, and has elevated the role of community scientists. So Matt's scientific accomplishments are really easy to list. Well, actually, they're really hard to list because of how many there are. But Nick and I wanted to share a little bit more about Matt's personal impact on us as scientists as well. So Matt, I first interacted with Matt as an undergraduate. I was obsessed with these little blue butterflies and what plants they were choosing to lay their eggs on. And I've been poring over Matt's papers on how host plants could affect insect evolution. And I ended up crossing paths with Matt after driving out to Nevada with a car full of host plants, these sluggy little caterpillars I was trying to get measurements on. And meeting him truly felt like meeting a rock star. I'm sure does not even remember this. It's totally
I'm manufacturing a memory as you're talking. Yeah.
So I remember thinking, wow, like these names on paper that we read can really be connected to kind, accessible people, and all that science talk can be underlain by a deep love of bugs, which I also had at the time, still do. So those early demystifyings and humanizings of scientists can be really important, I think, for all of us. And that was one of those for me. So thanks, Matt.
I was working with Matt on my PhD committee. I still actually now I tell everybody you were my co-advisors because of how long we basically spent together.
Nice.
I was always if there was an inch of Matt's door open, I was popping my head in and saying, hey, can I show you these statistics? Can we talk about this? And I'm sure he was really busy, but Matt's the kind of guy that he would take that 5 minutes and still take the time to at least humor me as he went through that. And you'll see this in the talk. Matt is amazing at taking some really complicated statistical ideas and explaining them in a way that everybody can understand. And he also loves bugs in general, insects. And so not only looking at the statistics, but also kind of trying to understand why and what are the consequences of it. Like me, Matt took some time to find biology. He was actually spent time in the Peace Corps in Ukraine trying to become a professional writer and a poet. I think he does have some poems actually out there. Probably also one thing when you read Matt's papers, it's super easy to read. Some of the best papers I've read. And that's probably a big part of it. Unfortunately, his aspiration to earn money through writing didn't pan out, ended up coming back into biology. I do remember something he told me that was like, I was interested in looking into research as a PhD in English and literature or history. And it was like basically all they do is critique others that have already been critiqued. So there's not a lot of opportunities. They're like novel ideas, and that's kind of one thing that brought them into biology.
Oh my God, Nick, that's terrible. You shouldn't say that in public.
Announcements, don't forget to do this afterwards. There are questions that you need to submit to the Q&A tomorrow, and I think there's a link in the email, so don't forget to do that before tomorrow's class. So
Oh my God. Nice, good. Thanks, folks. I feel super honored to have a double introduction. That was way too kind. Thanks, Nick and Mariah. Super nice to talk to folks. Really happy to be here. I was here, I don't know, we were just trying to remember like 7 or 8 years ago. It's always been a great place, but I feel like energy going on now seems like really at a high point. So really congrats to all of you, and looking forward to talking to people tomorrow. So I'm going to tell you about work we've been doing trying to understand insects in the Anthropocene. And I like to admit up front that none of this stuff is really what I meant to do with my career. I started out being interested in other things, speciation, even through postdoc, which was really classical quantitative genetics. I was headed in different directions, and I kind of got shifted from thinking about where species come from to thinking about where they're going, which is extinction, for I think obvious reasons, as you'll see. But I actually want to start with extinction. You've all seen graphs like this. This is variation in extinction rate through time from some group of marine invertebrates. These graphs are always changing a bit as paleontologists improve our understanding of the past. This is a pretty recent one. I like this one because it makes the past look a little bit messier. There probably weren't just 5 mass extinction events in the past. We're still trying to figure that out. There's lots of things we don't know about extinction. We don't really know how resilient different ecosystems or lineages are to extinction. We don't know things like whether or not population level stressors also lead to species level extinction. Unfortunately, we are living in a time when it's becoming more and more possible to study ongoing extinction. Anybody know what that bird is? So that was the last of the Hawaiian honey creepers, which as far as I know is the first extinction at a higher taxonomic level. So that's the last of an entire family gone. If you go to YouTube, you can find the last recorded song of it. It was just declared extinct by the U.S. Fish and Wildlife Service last year. So this raises this important issue of whether or not we are in a mass extinction event, which is not a simple question because paleontologists study big chunks of time and we study really narrow slices of time. So there's sort of a units confusion. Also, we study species or populations and paleontologists study higher taxonomic levels. I think the answer is probably yes. This is one take on it, again, recent that I think, or fairly recent, that's really good. These folks asked, if we were in a not in a mass extinction event, in other words, if we assume background extinction rate, you know, the troughs from that graph up there, how many years would it have taken to lose the number of species that we have lost in the last 114 years? And the answer is, well, for example, for fishes there, if we were not in a mass extinction event, it would have taken 2,000 to 6,000 years to lose the number of fish that we've lost in a little more than 100 years. So we're in a mass extinction event. That's kind of abstract. The numbers aren't abstract. And apologies, I'm starting in a pretty dark place here. It'll get slightly less dark, but not much. So the numbers are more concrete there. There's numbers of species lost since 1500. We have a really good handle on that for mammals, and for birds, 140 is probably a little bit more than that. We don't really know for plants. People are trying to work on that number. For insects, we don't really know at all. I'm going to come back at the end to this issue of how we're keeping track of current extinction rates with kind of an advertisement for an ongoing or a new project that I want people to know about. So I'm going to get back to that idea of ongoing extinction, but for now, what do we want to know in sort of a conceptual sense about extinction? Here's 3 questions for you. So what are the big stressors, and to what extent are they context dependent? This is kind of what the public wants from us, and we're all publicly funded to one extent or another. So it's like, you know, what do I have to, should I really worry about pesticides when climate change is happening, or vice versa? Important questions. What are the mechanisms involved in population level stressors? And you might think it's weird to separate that question from the first one, but I actually think that's useful because it's often the case that we can identify stressors with confidence even when we don't understand the mechanisms. And when we talk about conservation and advocacy, keeping those separate has actually been useful. And again, this gets back to where I'm actually talking about things that I sort of never meant to be involved with in terms of advocacy, and I'll use that word again later. And then can we forecast species-specific outcomes? And this will get back to some big sort of history of life questions. I'm going to talk about butterflies because I'm a butterfly biologist. We hope that butterflies are somewhat reflective of insects. And I'm going to start by giving you very current report card on the status of butterflies across the country. So this is the output of a recently concluded USGS-funded status of butterflies working group that I was lucky to be part of, where we took all multi-species monitoring programs across the country. You can see the tally over there, and just asked how have numbers changed over the last 20 years. And it's worth saying at the outset here that this project starts in 2000. And I actually think that's never early enough to actually know the depths of declines for the Anthropocene. So I'm just saying that now to remind myself to get back to that. But what we found is a 22% reduction in the overall abundance of butterflies flying around the country over the last 20 years. And that might sound extreme. There's a lot of variation among regions, which you see over there with the Southwest and the mountain prairie. And actually in this map, Utah is mountain prairie. Those are the most severely declining regions. These rates agree with a couple other lines of evidence. We've done work with sort of subsets and slightly different data sets in the West and got nearly identical conclusions. And more important, I think, that number of 22%, that comes from an annual compounded loss of like 1 to 2%, which sort of eerily keeps turning up from lots of different insect data sets in different parts of the world. So this really does seem like a generalizable decline in the density of insects flying around. In terms of individual species, there's about 10 species declining for every 1 species not declining, which is what's being tallied up there with that histogram. So that's percent change, sort of individual species level percent change, and then counting up the number of species. You see the red bar is a whole bunch of species in all different parts of the country that are declining over the last 20 years. So unfortunately, we have lots of material to work with if we want to understand the stressors of the Anthropocene. Okay, let me now zoom in to the part of the country I know. This is the Shapiro Monitoring Transect. That's Art Shapiro, my former PhD advisor at UC Davis. This is the transect that Art set up. This is across Northern California, Sierra Nevada, Central Valley, Coast Range. The year next to each site is the year which continuous monitoring began, and continuous monitoring means the sites have been visited every 2 weeks when there isn't snow on the ground since the date there. So we're at 53 years of data, and Art continues to collect data every 2 weeks, essentially all year round at these low elevation sites, which, let's face it, is kind of nutty. As far as I know, that much continuous data collection by a single observer has no other precedent in science. And I always say that hoping someone will come up to me after a talk and give me another example, but I haven't found one yet. 53 years of biweekly data collection is a lot. Out of my lab, we've started collecting the data up here because Art's knees started to give out, but he's still doing that. Lots of valuable things here. You might know the British Butterfly Monitoring Scheme is the world's other really old monitoring program, and they have more sites and lots of different observers, but what they don't have is a severe elevational gradient like this, which is super useful because this puts relatively pristine sites in at least sort of geographic proximity to very stressed sites. So things we've learned, here's the pattern that really got me involved in the data. So that's a sort of step change in the butterflies at low elevations in the mid-90s. So I mentioned this wasn't what I meant to do with my career. So I joined Art's lab in the very early 2000s, say 2000. So if you put 2000 on that map there, you can see it's very shortly after this step change. And I was sort of aware of Art talking about this, but I was doing other things. But in fact, even in 99 and 2000, whatever had changed for the butterflies was sufficiently dramatic that Art changed his census method. So it used to be the case every 2 weeks that he would record presence absence. So it's repeated presence absence sampling, which is still a lot because there's 160 species. But the decline was sufficiently severe that he started taking full count data. So we have counts of individuals at the low elevation starting in 1999. Any event, so I'm a grad student in the lab here, not really that interested, like doing other things. And then I move away for a postdoc. I come back 2008 in here. I've got a faculty job. I'm still in touch with Art, working on various projects, other things really, and I know that this is still going on. And finally, I find myself sort of unable to look away. Like, I got to figure out what this is, what's going on, partly because, I mean, obviously it's probably just of importance scientifically to understand this, but also like, these are some of the places that I grew up, is near these spots. So I felt sort of a personal responsibility to figure out what was going on. If anybody's driven the I-80 corridor through the Central Valley, you know it's a complicated landscape. Here's a picture of land use in the 3 counties that encompass those 4 sites. Green is ag. It's a super productive ag region, of course. That fuchsia, or whatever that is, that's the sprawl of Sacramento. There are little bits of other in there, the sort of green areas. Ag is not inherently bad for butterflies or other insects. Historically, ag in the Central Valley had a lot of productive margins that supported a lot of pollinators. So when I tell you about that decline in the 90s, you might think about the sprawl of Sacramento and pavement. It is the case that pavement is expanding. That's the conversion of land. So that's the rate at which land is paved over, sort of any open space, including ag, succumbing to urban sprawl. It's going up, but you see there's no particular step change in the 90s. As far as I know, the only thing that changed dramatically in the mid-90s was the introduction of neonicotinoids to the region. And I'm going to spare you some of these analyses because this is old and I want to get onto other things, but it sets an important stage. Neonicotinoids, neonics for short. I was asking grad students at lunch how many people know this, and I think it's always mixed. But if you don't know, you should know. It's currently the world's most popular class of pesticides. Clever chemists took nicotine, which of course evolved as an anti-herbivore chemical, morphed it into lots of flavors. But then the sort of the important part about it is that it's systemic. So farmers can spray it or get it onto their crops and the whole crop becomes toxic. In the Central Valley of California, it's very frequently used as a seed coating. So seeds are dusted, you plant the seed, and as the embryo emerges, it acquires the toxins and the plant becomes toxic, which is bad enough for insects, but a majority of that seed dusting actually blows off of those fields and ends up in marginal areas. The neonics are the colored lines there, and that's a log scale. So really ramping up. It's funny, a couple of weeks ago, there was, well, it's not funny really, it was actually kind of, it made me sweat. There was a IPM, so Integrated Pest Management Conference for the whole Western US in Reno, and they invited me to give the token like butterfly conservation talk. And I was given a version of this, and at the point that I was like pointing the finger at neonics, right? Because I'm like, you know, it's historical work, right? It's not an experiment, but I think it's very powerful. The room became so uncomfortable. I've actually never seen this happen. Literally, people were not keeping their butts in their seats. Like they were just shifting. And I paused here. I'm like, hey, maybe we should stop for questions right now. And then basically for the entire rest of the hour, people shot up their hands and they're like, I bet you didn't know that this changed in the Central Valley. And they had this long list of like ways that farming practices has changed, you know, like the ability to laser level fields, that evolved in the later 20th century. It was all fascinating. None of that changed in the mid 1990s. So I was like, okay, folks, this is still the best I got. This is my hypothesis for what happened to the Central Valley butterflies. Of course, it's not the only thing. We've done analyses of that, and the analyses suggest that the rate of pavement covering open land is of comparable importance to the introduction of neonics. Okay, but that raises the question of where butterflies are encountering neonics on that complicated landscape over there. I have zero toxicology background, right? I'm not even really a good chemical ecologist, let alone toxicological background. So at this point, I turn to the toxicology literature to just ask, hey, what do we know about the accumulation of, for example, neonics or any other compound in marginal ag spaces? And I was pretty disappointed. Hopefully there aren't any toxicologists in this room for multiple reasons, but it's a pretty thin literature. I mean, there is some research from parts of the East, from Europe. It's often been bee focused, so looking at pesticides and nectars, for example. But in terms of butterfly host plants, which of course lots of insects eat, very thin literature. So we figured it needed to get done. I stumbled into a nice collaboration with the Xerces Society for Invertebrate Conservation around that time, set up this sampling scheme. That's a bit of the Central Valley. The red is just diversity of pesticides used. Apologies, there's no legend, doesn't really matter. The dots there are spots visited that were ag margins, wildlife refuges, and some urban sites, just going to these spots and sampling only milkweeds as a focal plant. You got to start somewhere, right? And this was in 2018-ish, which was when the Western monarch population was hitting all-time lows. It was around 98% reduced density over historical highs. So it was a moment when people cared a lot about monarchs. go out and sample monarch host plants. And it mattered to sample monarch host plants in this area because it's a really important early spring breeding ground, right? So the simple question is, are they eating contaminated plants? Go collect the leaves, send them to a chemical ecology lab at Cornell, get this nasty graph back. Apologies, there's a lot going on here, but it's the gestalt that really matters. The rows here are compounds divided into herbicides, fungicides, insecticides. The columns are sites, and then there's multiple plants sampled per site. So a total of 227 individual leaf samples came back, everywhere from wildlife refuges, protected areas, to urban gardens. There wasn't a single uncontaminated leaf, and an average of 9 compounds per leaf. And what's worse is that more than a quarter of the plants had concentrations of a particular compound, chlorantraniliprole here, which you see with the white dots up there. The white dots indicates exceeding a known LD50 for monarch caterpillars. So a very toxic landscape, which was pretty interesting because the Central Valley is a landscape where I've driven with entomologists who say, all these conservation biologists worrying about the monarch. There's tons of milkweeds. Look around you, right? There are tons of milkweeds. They're 100% contaminated, and a quarter of them are downright toxic. And keeping in mind that the vast majority of compounds there, we have no idea what they do to monarch caterpillars because they've never been tested. At the time we did this study, there were a total of 8 compounds that had been tested on anything other than a honeybee, which is part of EPA regulations, which is to say we don't know. And there's lots of reasons to think that caterpillars won't respond the same as a honeybee. Since then, there's now about 12 or 15 of these compounds that have been tested on anything other than a honeybee. So that sucked us into this dark road of pesticide work. It's amazing that people keep giving us money to do this because, like I said, zero toxicological background. Don't worry, I'm not going to tell you about all these things. If you're a home gardener and you buy stuff from nurseries, as you would do as a home gardener, ask me over a beer later and I'll tell you the darkest story. But I'm not going to do that now. I'm going to tell you some brand new results, unpublished. So this is, again, a nice collaboration with the Xerces Society. Asking about host plants in urban areas. Why should we care about urban areas? Obviously, we live in urban areas, but also, if you're interested in insect conservation, you've probably seen the literature where green spaces in cities are often being put forward as like bioarcs, right? That like as mega cities expand, we should think about the insects that can live with us in these spaces. It's a nice idea, but it means we should look critically at what these places are and can insects use them. So really, Xerces Society did the organizational work here to get their network of amazing volunteers to go to green spaces in Albuquerque and Sacramento, both private yards and city parks, and just walk into these parks, have a regional list of butterfly host plants, and just sample host plants. So this is very descriptive, but there are questions. Questions were like, what's going to be more contaminated, woody plants or herbaceous plants? Things we might want to know if we think about these places as bioarcs for insects. Also, how about private areas versus public areas in terms of contamination? Here's what sampling looks like. That's not very easy to see, apologies. That's a map of Albuquerque. The circles show the genera of plants sampled there. So very non-random sampling here, not a design you would come up with. And they're zooming into one park. Again, this is just private individuals walking into a park following directions to find butterfly host plants. You could see some breakdowns of insecticides, herbicides, fungicides. You can probably guess that I'm working towards another mass contamination story here. Here's a picture of prevalence. So this is organized a little bit differently than the heat map. Compounds on the x-axis here, and then what's being counted up is just the instance of contamination. So in other words, this particular compound was found in this many of that plant genus, and then et cetera, et cetera. My chemist friends get frustrated with me when I make prevalence graphs like this, because this is ignoring concentration. In other words, here I'm treating a trace concentration the same amount as a heavy concentration. And I actually think it's kind of reasonable because we have no idea how those concentrations line up to mortality of these insects. So I'm like, you chemists, toxicologists need to do some work to tell me what those concentrations mean. Until then, I think the conservative approach is just to tally up occurrences. In any event, you can see Sacramento has a lot more going on than Albuquerque in terms of contamination. all kinds of fascinating stuff in here. Not a chemist, but you can just Google these things and learn fun quotes, actually miserable stuff. Like under fungicides, oh yeah, thibenicarb there, which is the tallest bar, basically found in all the plants genera that were looked at in Sacramento. It's a strictly rice fungicide, should not be in the city limits of Sacramento at all. But basically, if you're an ovipositing butterfly, you are going to encounter that in the city limits of Sacramento. And fungicides do have impacts on caterpillars. Or where's atrazine here? Oh, atrazine here. That's a really important herbicide in corn. What the crap? So if you Google it, you can read the Wikipedia page and the map. Like the eastern half of the country, the Midwest and Greens, is just a blanket of atrazine. Doesn't really look like it should be in Albuquerque at all. It is everywhere. It's also a restricted use pesticide, so it should not even be in private yards at all, but it is pretty much in every single sample looked at. Okay, but what about actual questions? We can compare herbaceous or woody. This is kind of like of just sort of basic biological interest, like what is the landscape insects are finding there. I expected more contamination in woody plants. In fact, it was a little bit more. So these are compound accumulation curves, right? So just like species accumulation curves, treating this like as the samples come in, you can ask how many more compounds you discover. There's a bit more in oaks and willows there, but actually those herbaceous plants are pretty contaminated too, which I was kind of surprised because the woody stuff being there longer, I would thought it would have accumulated more of these toxins. How about private versus public spaces? So private on the left, public on the right, Sacramento, Albuquerque. This was the biggest surprise. Nothing should surprise me in the toxicological world now. You can see these curves are not leveling off. So we really don't have a good estimate of the number of compounds in these spaces, but in terms of magnitude of contamination, they're on a similar scale. These private yards are 100% Xerces volunteers who are the most like bug-friendly, organic-y folks you could possibly imagine. Their yards are as contaminated as nearby city parks where people are actively out there in hazmat suits or whatever the heck spraying. Okay, that's the pesticide story. Let's move out of that. Let's get up into the mountains where we can at least forget about pesticides a little bit. Those graphs over there, let's keep my eye on my time. There's a lot going on here, but this is a summary of like 15 years of us understanding the Shapiro data set, so there's a lot going on. Each dot there is a population index that's averaged across all the species at a site in a year, put on a standardized scale, so positive values are better years, negative values are worse years, things you can see here. So that decline in the 90s, and you'll remember when I showed this to you before, you could see the step change because it went back to the 70s. It's kind of like a nice problem with 53 years of data that it's often hard to get it all into one graph. So boo-hoo, I know. But so this is starting here. You can see the downward trend and then a plateau. And then notice that interesting bit there. I'll come back to that at the end. That's one of like the kind of few bits of sort of bright spot in all of this stuff. More recent declines in the mountains. So you see that sort of more recent, more gentle downward trend. I was about to ask, does anybody remember what happened, but then I gave it away. 2011 to 2015, the so-called millennial drought in the West, when it was as hot and dry as it had been in 1,000 years. This was truly noticeable walking around the Sierra Nevada. Like there were just fewer butterflies in these places that we had been going, which produced a couple of kind of surprising results. So if you look at the comparative between the mountains and the valley there, where they're on a standardized scale, in terms of deviations from a historical baseline, it seems like the mega drought years pushed the mountains to somewhere near a half a standard deviation away from a historical baseline, which is pretty much where the worst times have gotten the valley to. Okay, so a couple of things there. What? That doesn't make much sense because the low elevations have been exposed to all the stressors of the Anthropocene, right? The pavement of Sacramento, pesticides, and climate change. The mountain sites, you know, there's always the issue comes up of pesticides that could go up into the mountains. We have not quantified that. For the most part, those sites are quite pristine, nice places. So it's really climate change. Okay, so in a minute, I'm going to try and convince you that actually climate change is that bad in the mountains. But before that, even consider the irony here. I would say this runs counter to global change biology theory, if you want to say there is such a thing, where I think before the mega drought years, if you would have told us, or maybe anybody in this room, that the valley has already suffered because they've been nuked by pavement and pesticides, and now you're about to be hotter and drier than you've been in 1,000 years. Okay, put your money down. Who's going to suffer more, the mountains or the valley? I think you would have put it on the valley. The valley's going to suffer more because it's already been pushed to the edge, and the mountains supposedly provide all kinds of ectothermic refugia, right? You expect these animals to be able to move up slope, right? That's what we've been telling ourselves for 20 years and find, cooler north-facing slopes, whatever. That didn't happen. Okay, so the question is why? Here's just a lead in to my explanation of why. I'm not giving the underlying analyses here, but those are coefficients relating nighttime temperatures to interannual fluctuations in butterfly richness across the elevational gradient. Warmer nights are worse for butterflies at high elevations. Okay, but that doesn't tell us why climate change would be worse in the mountains, does it? Except it does, because nights have been warming quite a bit faster in the mountains. You may or may not know this, depending on how good of a climatologist you are. I did not know this until I downloaded climate data and just made this graph and then started reading the climatological literature. So here's minimum temperatures from the 10 sites. Cooler colors are high elevations on a standardized scale. For the low elevations, you see some ups and some downs. This gets into the mega drought years here. But note the nights at low elevations had been moving up for quite a while. It was subtle until it wasn't. And then I think in the mega drought years, it really started to have a big impact. So this is where we want to think about mechanisms. Why would nights be bad for butterflies in the mountains? So it could be the case that warming nights are bad for ectotherms. It's been hypothesized, for example, that it's harder to recover from daytime heat stress if the nights aren't cool enough. And there's other ways that could be direct physiological stress, but it also could be effects through plants, right? This was the mega drought we were talking about. And when we were walking around the mountains in the mega drought years, we really thought, this has got to be a plant story. So there, I don't know if you can totally see, but on the left there, that's one of our big meadows at Donner at the height of the drought. It's pretty brown. And then here it is immediately after the drought. So I thought it was really going to be a plant story, which is to say we wanted to test this kind of a model. So in the lower right there, that's the butterflies you're counting in any one year. That's what we want to understand when it's a good year for butterflies, when it's a bad. And then the arrows are sort of causal drivers. So arrows going into that include weather that year. Is it a favorable weather year? Also lags effects of weather, and then weather that is mediated through host plants, right? This is like a basic model that I think we want to understand for insects and climate change. We don't have historical data on hosts and nectar plants. We've started collecting that for this project. We didn't have that yet, so we turned to productivity data or NDVI. Has anybody worked with NDVI data in here? I had not yet. It was full of surprises for us. I'm not going to give you all the results from this model because there's a ton. I want to just give you one NDVI highlight, and then I'll zoom in back to the butterflies. We found a consistent and really strong negative relationship between NDVI and butterflies, both same year and lagged. In other words, when you get higher NDVI signal of greenness, sorry, I didn't define that. It's a satellite-based measure of greenness, right? You get fewer butterflies. At first, we were like, come on. Can nothing be easy? Because we thought that'd be a measure of, it's a good plant year. That's going to be a good butterfly year. No. So turning to the literature, there's a couple of reasons that might be true. One, it's been found in other studies that NDVI reflecting greenness can be reflecting the greening up in a carbon-rich but nitrogen-limited world, and it could be that butterflies are eating less nutritious plants. I don't know. It's also been suggested, specifically in the Sierra, that higher NDVI values have reflected the overtaking of meadows by grasses and sedges and the loss of herbaceous plants. Peter is grinning. I'm hoping that means that makes sense. But don't say anything, Peter, if it doesn't make sense. Anyway, there's some reason to think that others have found a negative relationship between productivity and butterflies. It gets complicated, though, because there's also a pretty clear signal where plants in the mountains like cool, wet winters. If you have cool, wet winters, you have higher productivity. Higher productivity was associated with fewer butterflies. That sets up a negative indirect effect because a positive times a negative is a negative. more complicated, and apologies here, I'm not showing you coefficients or detailed results. I'll show you some coefficients in a second, but a pretty interesting balancing of factors here where we can estimate a direct positive effect. Butterflies in the mountains like cool, wet winters. And then we sort of knew that observationally. And that effect is slightly stronger than the negative indirect effect mediated through productivity. Okay, what do we do with this? Well, on the one hand, regardless of the complexity of NDVI, I think we're at least controlling for something about what plants do. I'm not pretending that we're doing everything, but we're trying. And then we really confirmed the fact with these analyses that winter conditions matter a lot for butterflies in the mountains. So let me zoom in now and just look at a simplified bit of results here that'll get us, I think, to a mechanism about how climate change is impacting butterflies in the mountains. And here is going to focus on one site. So this is Donner. In that previous analysis I was just talking about in a qualitative way, we were using one of these downscaled climate models, and I had my suspicions about the snow data from that model in a topographically complex landscape. But for this one site, the Central Sierra Snow Lab is literally at our site. We walk by a Central Sierra Snow Lab and count butterflies. So it was a good resource for getting the highest precision, I hope, snow data to line it up with our butterflies. We did a bunch of preliminary work to figure out how to tally up that snow data in a way that effectively explains some variance for butterflies, and then line up these factors. And this is our hierarchical Bayesian action. I'm going to show you results at a species level and then across species parsed into overwintering groups. We had a strong suspicion going into this that if there was going to be any mechanism for winter affecting butterflies in the mountains, it would be mediated by how you spend the winter, right? And temperate zone butterflies spend the winter in all of these different ways. Here's results there. I know there's a lot going on here. Note first that that productivity relationship there is mostly negative. I don't want to dwell on the one that's not negative there, although it's probably interesting. We get a negative effect of minimum temperature. So this is recovering with totally different analyses, something we'd seen before, that warm nights are bad for butterflies in the mountains, particularly in winter, and a positive effect of April 1 snow, and a pretty strong interaction, which looks like this. So here's the relationship between minimum temperatures and April 1 snow on a standardized scale versus the probability of occurrence of any particular butterfly. So this has been estimated hierarchically across all the butterflies that live there. The red dot represents the conditions at the depth of the mega drought years, where you can see the combination of min temps and April 1 snow push the probabilities lower for seeing any one butterfly. I suspect our hypothesis is that this reflects a mechanism whereby when you have less snowpack and you have warm nights, things that are overwintering, especially in earlier stages like eggs, are just not as insulated by the snow, and they're probably waking up and like trying to come out of diapause in the middle of winter and maybe just burning reserves and not even being able to, eat your way out of the egg when spring comes, or possibly even hatching too early and just being long dead by the time spring comes. It's funny, for a while I presented this and I would say, and we can use this to inspire experiments. Actually, the more I think about this, and this being estimated across 100 and some individual species and sort of topographic complexity, I'm not actually sure you can do that in an experiment. I know people you can push back and say, surely you can set this up in a growth chamber. I don't know, frankly. I actually kind of think this is the mechanistic result from observational data. I don't think we can do the right experiment. We can look for agreement with other analyses. We also did some work that I'm going to spare you almost all of, working with this data. This is North American Butterfly Association counts across all these sites across the West. This is publicly available data. Folks go to these spots in the middle of summer, count all the butterflies in an area. We've used this to characterize where butterflies are declining more severely. And then we asked about the climate in those areas. And we found a messy but believable signal that in places where fall is warming more rapidly, you're seeing fewer butterflies. That's just raw data. It's not cleaned up at all, not accounting for spatial autocorrelation or anything. Model output would be fancier looking. This is the difference between fall and summer warming, and fall is actually warming. Seasons are warming heterogeneously, and fall is warming more rapidly the farther you get into the US Southwest. And places where fall is warming more rapidly than summer are where you're seeing fewer butterflies, which is on the y-axis there. I think this is actually complementary to the winter result. I think the winter result points to overwintering as eggs and early instars. I think this, which was fall warming, points to the end of the growing season as kind of an Achilles heel for the arid West for insects, where insects, if they're still active, are either trying to take their last bite of food or they're adults trying to fly around, find the last bit of nectar in order to lay eggs. And as fall, end of summer into fall has gotten hotter and hotter, like we're experiencing right now, I think those resources are drying up, which means you get fewer butterflies the next year. Let me wrap up this section, and my third section will be relatively shorter. It'll be okay. Returning to this model here, not pretending like we understand everything about plants, right? Like I said, we are collecting nectar data now. Nectar data is relatively straightforward because you can see butterflies on nectar plants and you can census them. Collecting the all-important data about the health of host plants is super hard. We actually do know what host plants these butterflies eat, but like just because I know that a particular species of willow is used by a particular hair streak at a given site doesn't mean it's easy to go census that host plant there, because the butterflies will pick particular patches of host plants. What you have to do to parameterize this arrow is you've got to go out and figure out where the caterpillars are each year, which is a crazy thing to do. This is something that Nick and Mariah have done. I got this from my colleagues Lee Dyer and Angela Smolanich, who have been doing this for decades all over the Western Hemisphere now. Basically, you have to get teams of people out there and search for caterpillars, and you do this year after year, and you build up caterpillar data, and hopefully someday we'll be able to really parameterize that model. Something else I was going to say here. Yeah, so we're doing this every 2 weeks now, which as far as I know it's sufficiently foolish that I don't think it's ever been done before to monitor caterpillars adults at the same time. But just for this talk, actually it's funny, I had not yet, even for the proposal, and Zach is implicated in this proposal, I had not taken the historical caterpillar data and put it together with the adult data, not even for the proposal, which I probably should have done, but frankly, I didn't think it would line up. But getting ready for this talk, I thought, okay, I'm going to finally just like crack that open. And so I took the caterpillar data just for the whole Eastern Sierra, because historically, that's where much of the collection had gone on. We're now doing West Slope stuff too, but just Eastern Sierra caterpillars. And one of our butterfly sites that's on the east side of the Sierra, count up the total caterpillars found per year, make it into a standardized insect, and then I have a way of making an index of how good a butterfly year it was. Put those suckers on the same scale, and dang, at least they do something together. I was pretty stoked. I mean, here you see the mega drought years, and then coming out of the mega drought years, yeah, I mean, it's not rocket science at this point, right? It should be the case that caterpillars suffer when adults suffer. But suffice it to say, it didn't have to be that way, because you could have situations where caterpillars are really doing well in a year, but it's the nectar plants that suffer, and you could see a disjunction between these things, or vice versa. It's interesting to see them doing the same thing. There's a crappy correlation between butterflies in one year and caterpillars in the same year, but then what if I lag it? So here's butterflies in the previous year predicting caterpillars in the next year. Now I know I just spent $600,000 of NSF money to recreate the butterfly life cycle that you learned in 6th grade, but that's science, right? It's true. Adults produce caterpillars in the next year. Okay, I do think that kind of sort of like, sort of crude index across many species is useful for figuring out mechanisms. But let me wrap up here by getting back to history of life stuff. An important question I think about extinction in the history of life is the extent to which extinction is random or not. Mariah mentioned, because Mariah asked me in email, how did you go from English major to science? I confessed that the Stephen Jay Gould's book, Wonderful Life. Has anybody in here read it? A couple people. I hope it still holds up. So I read that book long before I had gotten back to science, and was really intrigued by his hypothesis that there's like background extinction that happens that's Darwinian, that like some things are becoming better adapted and they're pushing others out. But then when the meteorite hits, it's a crapshoot and all rules are off the table. In other words, there's, predictable extinction here and then unpredictable extinction there. That's a pretty interesting hypothesis about the history of life. I think there's been a feeling in the contemporary extinction literature that people are able to predict it. I don't know if that's true yet. Apologies for that horrible looking graph. We're working our way towards this, and I'm not going to torture you too much with the details, trying to put together life history and other sort of species specific information for butterflies in the West. We're not really doing much of a better job than anybody in the insect world is, which is to say we can take traits like volatinism and wingspan and host range, and we can throw them up against the rate that populations are changing, or I'm just going to refer to this as risk, with higher risk being they're suffering more, and try to predict it. The best we get is like 10 to 11% variance explained, which is to say relationships look kind of lousy like that. So there's wingspan. So bigger butterflies are doing slightly worse. So higher risk index there means they're doing worse, lower insects are doing better. Like, you can get some associations. Variance explained is not good. And the next time you see a paper that claims to have found the correlates of decline, just drill in for the variance explained. I think it tends to be in the 10 to 12% range. At least historically, that has been true, which is to say, I don't think we're answering Gould's hypothesis. And at least for butterflies, many of the correlates that we have found tend to go away when you correct for them phylogenetically. Here's a phylogeny of butterflies in the West. Here's nymphalids, which are your big butterflies, monarchs, and other things. The big butterflies are suffering less. Is that because they're big or for some other reason that we haven't measured? That's where the phylogenetic correction comes in. I think the problem here is actually not that we need better trait data. I think we need more studies at finer scales. I'm going to sort of work towards a wrap up here by just telling you a population level story of how traits matter at populations, and then I'll be almost near the end here. So you remember that upswing in the Central Valley here? So this was the mega drought years. This was where I said if someone had asked you to put your money down on who was going to suffer more in the mega drought, I think you would have bet on the Central Valley. Central Valley actually weirdly had some good butterfly years in the mega drought there. We started by throwing all the usual traits against this, didn't find much. I had an idea that maybe association with agriculture would have explained something, right? Because it was a known thing that farmers, in many cases, were sort of overwatering in those years. We tallied that up in a way I don't want to bore you with, was not association with agriculture. We found that these two sort of complicated traits were really informative. So here's change of date of first flight. Butterflies, like most insects in hot and dry years, will start earlier, right? They'll emerge earlier and get going earlier. So over here is going to be populations. Actually, this is going to be a graph of species at these few sites. Over here is going to be species that emerged earlier during the mega drought years. And then on the y-axis is going to be change in abundance, all during the mega drought years. So really zooming in on those years just for the low elevation sites. It's a negative relationship such that species that emerged earlier in the drought years did better, and that was driven entirely by volatinism, where it's really the multi-voltine species that were able to emerge earlier and really simply just take advantage of the extra demographic time for it to increase and have some good years during the mega drought years. I don't think it's terribly profound, it's demography, although it does challenge yet another simple expectation in global change biology, the so-called mismatch hypothesis, such that if specialized insects, and these things are all pretty specialized, if they advance their phenology, it's going to break their relationship with host plants. It's patently not true for the butterflies that we know. They can shift up to a month early and they find something to lay eggs on. They're pretty good taxonomists. They find something closely related enough that's going. So not only did that phenological shift not break that trophic connection, but it gave them some demographic power to actually have some pretty good years. It didn't work for very long, right? They crashed again, right? This isn't all a rosy story. But the opposite is true of the mountains. And this is the last complicated graph I'll show you. Moving up to the mountains, take all of the population dynamics there. What I'm going to show you is a constrained ordination where it's taking all the ups and downs of the population there and trying to explain that multivariate mess with every sort of possible life history trait we could think, which, is just sort of like throw it at it and see what'll stick method. Here's the mess of ordinated population dynamics. The color coding there is population trajectory over 50 years. You see darker points at the top. Those are species that are doing well in the mountains. Lighter points at the bottom. Guess who's doing badly in the mountains? The multivoltine generalists are sucking it in the mountains. Flip-flopped 180 degrees from the low elevation where the multivolting generalists who can shift that trophic interaction if they move a month earlier are doing well. Being a multivolting generalist doesn't work in the mountains for reasons that are not as obvious to me as the opposite pattern. I think I understand what's going on in the valley. I think this is more complicated. If you are used to having multiple generations in the mountains, somehow it may be the case that you're getting truncated at the end. I'm not that confident in that. But it's also the case that a lot of these species have historically had some demographic input from low elevations, and I think moving through that matrix has become harder in recent decades. It's complicated, but the point is that it doesn't actually work to be a multivolting generalist up there. And with that, I skipped a transitionary slide there because I want to make sure people are awake for my last couple messages here. Not pretending like we understand the correlates of decline in butterflies. I don't think anybody does in insects. In fact, even I could for sure make some enemies now, but the historical literature for mammals and birds looking at correlates of decline, like I don't get anybody looking at me evilly yet. A lot of those studies were based on like red list data, where some of the same traits that put things on the red list were then used to predict their position on the red list. There's a little bit of circularity going on there. It doesn't mean it's not real, but they're tricky analyses. I don't think we have the answer to Gould's hypothesis about extinction, but we can for sure put our finger on who's going down. That's the last thing I wanted to note, which is partly an advertisement for grad students. If you're interested in the biology of extinction, there's lots to work on. This is the West Coast lady, Vanessa Annabella. I think it should be the poster child for insect conservation in the West. Here you see various time series going down. This is an animal that our montane sites used to be seen like 60, 70% of the days you're out. Now we see 2 or 3 in a year. This is another species where you would not have put it in the bin of likely decliner 20 years ago. It's a big thing. It's got multiple hosts, native, exotic. It's highly dispersive. Its range is basically all 11 Western states, and it is in a coordinated range-wide decline. Here's another one. The common sooty wing is not common anymore. It's going through sort of sequential extirpation in the Central Valley. When I was a kid in the Central Valley, this thing was so dirt common in urban lots in Sacramento, we called it a moth, as if that was some kind of an insult. Like kids, like it was just a great thing. It's actually pretty charming though. Going in decline. These things are in decline, a couple of marbles. That's been petitioned to be listed at a couple of different levels recently. Not everything is declining. I don't love the phrase winners and losers because the game ain't over, but there are some species doing better than others. That's the Western tiger swallowtail, which I'm sure you have in Logan. Does really well in urban areas, laying eggs, for example, on ashes and cottonwoods really high in the canopy. So that's a pretty interesting species if you're into this kind of thing. I'm going to skip my summary slides because they're super boring and end with my sort of advertisement for this last project that I wanted to get back to here. So I had started in this dark place here. We're not doing a very good job keeping track of these things. We really only know for charismatic vertebrates, which I would say is absurd because the idea that we're in a 6th mass extinction, if true, is the dominant ecological pattern of our times. We should be keeping good track of the species we are losing. So here's my idea, and this kind of sound like a stereotypical thing for a middle-aged professor to suggest, but I think we do need a new journal for this dedicated to the record of lost species. So the word lost here is kind of interesting because like if you've read the literature on the ivory-billed woodpecker, it's really hard to agree on extinction, right? It's actually not hard to agree on something being lost. You know when your car keys are lost, you can't find them. A species being lost means you keep sending people out for it and you cannot find it. There are lots of species that are in the lost category. That number for the number of extinct birds, that's not the lost ones. There's a recent paper in the last couple of years that was published it was one of the Frontiers journals that estimated some 100 and some species of birds are in the lost category. No one's seen them for the last 10 years at least. It doesn't mean they're gone, but we don't know where they are. Problem is we don't have a unified place to collect this information. And an irony of this is that when you think about papers of the biodiversity crisis, papers on decline, species-specific information is often obscured into the appendix, right? because it's an important issue. And so those papers will often go in high profile journals where the centerpiece figure is a line going downward, right? And it's powerful because it's averaged across some hundreds of species. Okay, but the individual species for which we don't know where they are, they might not even be listed in the appendix. I think that's bad. I think moving forward, we need to do a better job of keeping track of that species-specific level. So we're going to have various article formats where you can report on individual species, you can do syntheses for entire lineages, and this is not just insects, this is the whole tree of life. We are working with the American Museum of Natural History to make this happen as a nonprofit endeavor. We're hopefully going to have an AI-based translation system, so people are going to be submitting in languages and all kinds of journals. I don't have any idea how that's going to work. But I'm super excited, and I think there is a bit of hopefulness here, right? Because like you can find your car keys again. There's an organization called Rewild. I don't know if you've encountered this. They're funded by Leonardo DiCaprio and some like super fancy people, where they've been funding expeditions to like go to remote places and look for lost birds and mammals, and they've found some. Not a lot, but they have found some. So you list things as lost, people can find them again. That's some hope. And at the very least, we keep a record of, I think, the most important ecological pattern of our times. And with that, thanks to lots of folks. And it's good I talked less than an hour. Okay, I'm going to stop. Thanks. Yeah.
So great talk.
Yeah.
I was really
Thanks.
curious also about whether or not, so in addition to facing census population declines,
Yeah.
has there been any work done to see whether or not there's reductions in genetic diversity across populations, reduction in effective population size, and if that is or isn't necessarily correlated with census population?
Yep. We have not done that. It's something we should probably do. Yeah. So let's talk about it more because you are probably connected to somebody who knows how to do that. So yeah. Yeah. I'm into it. It's important. Yeah.
Change across everything.
Yeah.
Like a really complicated, messy story.
Yeah. Yeah.
With those species-specific graphs that you showed later, I was wondering if there are any clearer trends where, okay, this species is declining because of this one thing.
Nope. And that's where it really takes sort of multivariate storytelling. And I don't say that in a bad way, but we can pick out some of those species that are doing well, like the Western tiger swallowtail, and be like, okay, it's big and dispersive. It has native hosts and exotic hosts. It can lay eggs really high in the canopy. Maybe that lets it persist in urban areas where its hosts get access to water, but they're above the toxicity that's clearly floating around in agriculture. So like we can generate species-specific hypotheses, but in terms of picking out patterns from the data, I mean, I'm not saying there aren't any, but I'm skeptical of them because the variance explained is low. Yeah. Yeah.
Talked about volcanism and warming autumns. So I was curious if you have thoughts about the lost generation idea where the seasons might extend and then multiple species will try a generation at the end of the season.
Yep. Thank you for asking because some words came out of my mouth before and I cringed because I didn't mean to say that. I actually don't think the lost generation hypothesis is panning out, and it's been tested. I think it was with the Ohio butterfly data set where they've done a lot of work where they specifically looked for it and they looked for signals of partial generations and then asked if that was associated with downward trajectories over time, of which they've gotten a lot. And the answer was no. And I think that's true here. And for the multivoltines suffering in the mountains, I actually don't think it's truncation. I think it's more that they are relying heavily on some nectar resources in the second half of summer. And I think those are suffering. I don't have a very good I haven't done the quantitative analysis. Partly the sort of lost generation hypothesis has a cartoon of discrete generations, and most butterflies will have one early discrete generation, then an overlapping mess. I don't think there is one to truncate. So thank you for asking that so I could correct what I said before. Cool. Yeah, right. Yep. And no, and you know, there's parts of analyses from Europe, not just the British scheme, but other European countries where they've seen a signal of specialist suffering, which we don't see in our data, or in, I mentioned that USGS group across the whole country. Does not seem to be the case that specialists are suffering across this country and generalists are doing better, or vice versa. There doesn't seem to be a signal of diet breadth in North American data as far as we've got it. I don't know why, except I don't know, mostly, I mean, as you know, specialism, generalism is a local phenomenon really, right? So when we're doing these analyses like across the whole country, we're tallying up host range and saying this species eats 15 different plants. But in any one spot, it only eats one plant. So I don't think, I guess to answer your question, I don't think we know because the analyses have not often been done at the right scale. They've been done at these aggregated scales, which I don't think gets at population mechanisms. Yeah. Yep. Yeah. Hmm. That's a pretty serious toxicological question you're asking. All I know is we keep doing these snapshots, and each one is a snapshot that like encompasses different amounts of time. And we've done leaves, and we've also most recently used these silicone bands. Have people seen this method? This is new in the ecotox world, but it's good to know about if you want to dabble with it at all. I warn you, you get sucked in. So you remember those silicone like Live Strong bands? Remember these things? Okay, so you can take exactly those things. You can mass order them from whatever factory they come from somewhere. You give them to your chemist friend. They treat them. They seal them up. I don't know what they treat them with. They seal them up. You take them out in the field. You can hang them from a pin flag, and they become perfect passive absorbers of chemicals in the environment. And then you seal it back up. You send it back to your chemist friend. They treat it with something. The stuff runs out. You put it in a mass spec. So we did that in the Central Valley in a follow-up wildlife refuge study, integrating over a month. And it was the same miserable picture of just completely ubiquitous contamination. That study was asking if the center of wildlife refuges are refugia from pesticides, and they are not. There was no effect of distance from edge on contamination in wildlife refuges. Oh, it's miserable. So, you know, some of these things all have complicated lives, and some of them degrade into secondary and tertiary breakdown products that are worse than the primary one, which I say as if I know what any of those products are. I don't. But I think it's all bad. Yeah. It's a mixed bag. Yeah. Thanks for summing that up. It's good. Okay. Should we stop? Okay. Thanks, folks.